Well, good morning and TGIF. Here we are with quiz 5 right here. Good morning. The enzyme maltase catalyzes the breakdown of maltose. Ose means it's a sugar into glucose. That's another sugar. So maltose is probably a disaccharide, 2 sugar rings, and glucose is a 1 sugar ring. A technician added maltase to a solution of maltose. Maltase is an enzyme to be incubated at 37°C for 20 minutes. Samples were taken from the reaction tube every 5 minutes, and the glucose and maltose concentrations were measured. Which of the following is an appropriate negative control for the experiment? So to identify a good positive and negative control, you need to understand what the variables are in the experiment. So if we were to graph these data, our graph might look like this. The effect of maltase enzyme on maltose sugar. So the thing that's doing the affecting, you put that down here on this x-axis. So this is the independent variable. This is the thing that you're messing with. Independent variable. And then the independent variable affects the dependent variable. The dependent variable is the thing that gets messed with by you screwing with this right here. So as you add more maltase, you might expect the amount of maltose sugar to decrease. So you might end up with something like that. Yeah. Like if you add no maltase, then you're going to end up with tons of maltose, and you add a little bit of maltase and it eats up some maltose. And the more maltase you add, the more sugar gets eaten up, or actually the faster it gets eaten up. So anyway, if we were to do a negative control, what we would want to see is what is going to happen to the amount of maltose if there is no maltase there. So what we would want to do is the experiment the same exact way, but instead of adding maltase, we'd want to add something that's not going to mess with maltose, like water, for example. So instead of adding a constant solution of maltase, you could just add plain old water and see what happens to the maltose. Because maybe it's the temperature that's breaking down the maltose. We don't know. We need to do a negative control in order to see if it is in fact the maltase that's having the effect. So we would want to incubate maltose with water. Yeah. What's the best definition for the term hypothesis as it's used in science? A testable explanation for a scientific problem or question. That sounds good to me. An assumption based on a verifiable law. No. A comprehensive set of ideas explaining a natural phenomenon. Comprehensive set of ideas, that sounds like a theory to me. And a scientific model, no. So this is the best answer here. Testable. Yeah? Okay, and then you had to read this article right here, which was all about hormones in fish being affected by environmental factors. So they, well, I'm not going to summarize the article. You guys read it. If you have any questions about the article, let me know. Regarding the EPA's decision about controlling hormone disrupting chemicals, what might be some influences on their policy? Are they going to consider documented evidence of impact on wildlife over time? Yes. Are they going to consider documented evidence of impact in humans over time? Yeah, the EPA cares about both those things. Are they going to think about the effect on the economy? Yes, they are. Because if you're going to save a little like fish or something and it's going to completely devastate the entire economy, then maybe they're just going to let the fish die, because they do consider that also. Are they going to consider input from special interest groups, like, for example, the Sierra Club? which is an environmental group. Yeah, they're going to listen to what they have to say about it. Are they going to listen to maybe the paper industry who's discharging waste into the rivers? Yeah, they're going to listen to them also, because if the paper industry says, hey, look, if you don't let us use that chemical, we're going to have to use this other chemical and it's actually worse. And if you want paper, you're going to have to let us use that chemical because we have all this proof to show that we can't make it without one of these two chemicals. Yeah, they're going to consider that also. They're going to consider all those things. The EPA. Sumter and Jobling, that's the British ones, concluded that the expression of vitilogen in male fish was dependent on the concentration of industrial chemicals in the environment. Which of the following assays, an assay is an experiment to measure the amount of something, which of the following assays would most likely be used to measure how much vitilogen is in a blood sample? Okay, the key to you answering this question correctly is to understand the nature of vitilogen. It said in the paper that vitilogen is a protein. That's your key right there. Once you understand it's a protein, you need to choose an assay that's designed to measure proteins. PCR does not measure proteins. Spectrophotometry can measure proteins, but only at relatively high concentrations. And the concentration of this hormone inside fish blood is going to be so low that you're not going to be able to measure it on a spectrophotometer. Restriction enzyme digest, that deals with DNA. ELISA, booyah. ELISA is a super sensitive way to measure proteins with the use of antibodies. Booyah. That's what you got to know. To determine the specific estrogen mimicking agents, that means things that act like estrogen, which is a female hormone, causing the expression of vitilogen, researchers tested common industrial chemicals on the fish in the controlled environment of the lab. What variables might the researchers consider when setting up the experiment? The temperature of the water? Yeah, you don't want to just use a relative temperature. Age of the fish, it might matter. You might want to use different ages of fish. Concentrations of chemicals, that might matter also. You might want to use a little bit of chemical and a lot of chemical and everything in between. Materials used to construct the tanks, are they plastic? Are they glass? Are they metal? That might matter. So surely it's all the above. The hormone vitilogen is a protein found in female fish and is responsible for making egg yolk. That was the deal, is that these fish were making eggs in their balls, which isn't normally what happens, or their testes, excuse me. The expression of vitilogen is triggered by estrogen, which is a female sex hormone. They told you right there that it's a female sex hormone, and that's going to help you answer a question later on. Just taking skills. The fact that male fish are producing egg yolk protein suggests which of the following? Male fish have a gene that codes for vitilogen. Estrogen is commonly expressed in high levels in male fish. Something in the environment is triggering the expression of the gene for vitilogen. This is certainly a correct answer. This one right here, estrogen is commonly expressed at high levels in male fish. They just told us that it's a female hormone. So no, that's not right. Male fish have a gene that codes for vitilogen. Well, these male fish are producing this vitilogen inappropriately, which is causing them to make eggs in their nuts. So that's happening also. So that's happening and that's happening. So it's A and C. Yes? A little bit tricky. To test their hypothesis, Sumter and Jobling placed caged male trout downstream from other sewage plants. They observed that male fish produced egg yolk protein. Which of the following statements is the most accurate conclusion? British fish typically express male and female characteristics. That's a gross generalization there. The regulation of vitilogen is a Y chromosome linked. Is Y chromosome linked? That's the opposite of a gross generalization. That's a really fine point. And this study did not test anything about the genetics of these fish. It just tested how much of these chemicals they were producing. So although this might have something to do with it, that wasn't what the study investigated. Environmental factors affect expression of vitilogen. That is what the study investigated. And that was what they were able to make conclusions about. So when these studies, you have to make sure that you're not taking something that the study found and using that to make some sort of wild determination about something else. That's fine. You can make another hypothesis. Here's another hypothesis right here, but that's going to require a totally different study to test that hypothesis. We can't jump to conclusions in science. Based on the combined findings of Sumter and Jobling, the studies done in the US, which of the following conclusions were made? So they did studies in England, they did studies in the US. What conclusions can we make? Human activities are contributing to changes in the expression of sex hormones in fish. Yeah, seems like it. The estrogen mimicking chemicals will only affect carp fish. What? The ban on pesticide DDT should alleviate the problem. No, man. No, man. Yes. 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 Ah. Okay. How do you think we did? Think we got any of them right? Oh, we got 8 points out of 8 questions, so I guess we did pretty good on that one. Well, I hope you guys have a great weekend. It's Mother's Day. Snuggle your moms if you can. I'll see you guys on Monday. Bye.